Okay, so welcome to this next video in the playlist on voltage-gated ion channels. And in this video what we're going to do is we're going to look at the structure of voltage-gated sodium channels. Now we're not going to dwell too much on the structure of voltage-gated sodium channels because there's not really that much functional diversity in voltage-gated sodium channels, and there's not really that much structural diversity in voltage-gated sodium channels. Pretty much, a voltage-gated sodium channel is a voltage-gated sodium channel, and they're all pretty much the same. There are lots and lots of different types of them that have been discovered, and they're all localized to different places, at different parts of the body, to different cells of the body. And that's very, very important because it means that we can build pharmacological agents which will specifically attack these voltage-gated sodium channels at specific targets. But really they have very, very similar structures, and there isn't a huge amount to study in this, whereas voltage-gated calcium channels have a lot more that is interesting and is known. So we'll dwell much more on them, and potassium channels as well. But we do want to start with voltage-gated sodium channels. So let's start with the basic structure of a voltage-gated sodium channel. So voltage-gated sodium channels are made up of 4 domains. So if I start by drawing the cell membrane, and I'll put the channel here then, and the channel is made up basically of 4 cylindrical domains, which is a rough approximation. Obviously they're not perfect cylinders, but you can think of them as cylinders. So there's one, here is a second one, here is a third one, and here is a fourth one, and they go down and they form cylinders like this, so down at the bottom there, okay, and then you have another one down here, etc., like that. And basically the membrane obviously continues on down there, so this is the phospholipid bilayer here, so you can think of drawing these little diagrams for phospholipids if you want, etc. But this is the voltage-gated sodium channel pretty much here, and it's made up of these 4 domains. So we'll call this one domain 1, this one domain 2, this one domain 3, and this one domain 4, and there's a pore going down the middle of them, which is where the sodium ion will go. So when this opens, we can have a sodium ion here, which can basically go down that pore, so it can go in here, and it can come out the other side, basically. So that's the basic structure of the voltage-gated sodium channel, and this subunit, all 4 of these domains, belongs to a single subunit of the voltage-gated sodium channel. I should have said this. This is the functional subunit, and if you just have this bit, this bit with the 4 domains, then you have a functional voltage-gated sodium channel. Okay, so this subunit is called the alpha subunit of the voltage-gated sodium channel. So all 4 domains together forming this tube is the alpha subunit, and if you just have the alpha subunit, that forms a functional voltage-gated sodium channel. However, they can have stuck on their side basically an accessory subunit or an auxiliary subunit, which basically just modulates them. It affects them in some way, but it's not essential to be there, so it doesn't need to be there for the channel to be functioning, but it does affect the functioning of the channel, and this is called the beta subunit. So there are 2 subunits of the voltage-gated sodium channel, one which is an essential subunit, and it is basically the voltage-gated sodium channel. It's the channel through which the sodium moves. And then there is an auxiliary subunit which can or cannot be there. It doesn't really matter whether it's there. It does affect the properties, but the channel is still functional without it, and that's called the beta subunit, and together they make up a voltage-gated sodium channel. Now, we're going to study these 3 domains basically these 4 domains in a bit more detail. So off domain 1 comes the amino terminus of this protein. So you have, remember, this is a protein basically. This voltage-gated sodium channel is a protein. So it's made up of a great big polypeptide. So you start off with the first amino acid somewhere, and that has an amino terminus over here. And then you continue on with the amino acid structure. So you have this is the basic amino acid structure. You'll have some R group there, some hydrogen, and the basic amino acid structure is that you have a hydroxyl group down here. But of course, when you make a polypeptide, what happens is that you get the next amino acid along, and you form an amide bond. So this is another R group over here. So I'll just finish the drawing of the next amino acid. So you basically what you do is you can take off that hydroxyl group there, and you take off the hydrogen, and instead what you do is you bind the carbon with the oxygen, And the hydroxyl group and the hydrogen make water together, so it's a condensation reaction basically, forming this amide link. And basically you do this again and again and again and again, and you have absolutely loads of them in a great big chain. But at one end you will have an amino group, and at the other end, right at the other end, you'll have the final carboxyl group, which isn't linked to anything. And basically, domain 1 has the amino group coming off, and domain 4 has the carboxyl group coming off. And this alpha subunit is all one great big polypeptide. So basically, you start off here, you form the first domain, and I'll show you the structure of the first domain. And then it goes, the polypeptide continues on into the second domain. It then continues on to form the third domain, and then it finally comes into the fourth domain, and then finishes down here, basically. And both the amino group and the carboxyl group, they are on the intracellular. So this is the intracellular aspect. So they face inside the cell, they face the cytoplasm, rather than the extracellular compartment. Okay, right. So now what I want to do is show you the structure. So I want to show you, in terms of a polypeptide, the structure, how you form these 4 domains, basically. So now, if we draw it out in terms of what it is in terms of a polypeptide, then basically it forms like this. So you have a polypeptide, so this line is going to represent the polypeptide. So here is the amino group starting off over here, and then you have this phospholipid bilayer here. And basically, I'll draw the phospholipid bilayer going off like that. And basically, to form these great big cylinders, what you do is that the polypeptide just goes through the membrane, and then it comes back through the membrane again, it goes through the membrane again, it comes back through the membrane, then it goes up through the membrane again, it sort of bobs in a bit, sort of goes like that, and then it forms its final domain there. It forms its final membrane spanning bit there. That structure of the polypeptide going through in this exact pattern, it goes through the membrane, it spans the membrane a whole 6 times, that is all part of domain 1. So basically all of this forms that first cylinder there. Then, to form the second cylinder, you go on and do exactly the same thing. So domain 2 is exactly the same structure in terms of polypeptides as domain 1. So you continue on like that. And then, so this is the second domain here. So this is the second cylinder up there. So this is domain 2. So if I shouldn't have done it in orange, I'll highlight it in a different colour. So green, let's highlight it in green. So this is the second cylinder here. All of this structure from here onwards, all of that forms that second cylinder. All of this forms the first cylinder. So you can see how the polypeptide continues on from forming the first cylinder to straight into forming the second cylinder. Now, and it goes on basically. So each of these domains is identical. So the next one, the third one, has the same sort of structure. Well, they're not necessarily identical, i.e. the actual polypeptide structure might be different, but the overall way in which they span the domains, the fact that, it goes across once, it comes back, it goes across another time, so it's got its third membrane spanning domain, then it's got a fourth membrane spanning domain, a fifth membrane spanning domain, then this loop that tries to be a membrane spanning domain but doesn't get across, and then the final 6th membrane spanning domain. All that is conserved throughout, basically. So this is the 3rd cylinder there, and then I'll go straight in and draw the 4th cylinder. So the 4th domain of this protein. And there we go, there's the 4th domain. And then finally, it's going to end with a carboxyl terminal down there. So let me just divide this up. So this is domain 1 here. Domain 1, domain 2, domain 3, and domain 4. And you can see that their structure is incredibly conserved. The way in which they span the membrane is conserved. And basically you refer to this, if I label it on this one, you refer to this first bit that goes through the membrane in each of the domains as the first membrane spanning domain. So you call it S1. Then the next one, the second one, is called S2. The third portion of the polypeptide that goes through the membrane, so the third time that you go through the membrane in each one of these domains is called S3. Then the 4th one is called S4, and you're probably getting the hang of this now. The 5th one is called S5, and finally the 6th one is called S6. Okay, so that's just a bit of notation to label each of these membrane spanning domains. So this would be the first membrane spanning domain, S1, of the third domain. So the third domain of the alpha subunit of the voltage-gated sodium channel and the first membrane spanning domain of that third domain, basically. Okay, so this overall is the structure that makes up this voltage-gated sodium channel, basically. So each of these 6 membrane spanning domains together makes one of these domains, one of these cylinders, and together they all clunch together to make a pore in the middle. And basically the 4th membrane spanning domains, which is this one here, this one here, this one here, and where's the final one? This one here. They are very important. So all the S4s are very important because they have a positive charge. So S4 is positively charged, and these S4s all line the pore, basically, of the sodium channel. The orange, these S4 domains are the ones, oh sorry, no, that's not correct. S4 is the one that is voltage sensitive. S4 does not line the pore. It's S5 that lines the pore. So S4 is the bit that is voltage sensitive. So when the electrical potential difference across the membrane changes, something about the channel has to recognize that there has been a change in the electrical potential difference across the cell membrane and change the structure of the channel in response to that. And basically it's the S4 domains of each of these cylinders that changes because of the electrical, it responds to the change in the electrical potential difference and changes the structure of the channel. S5 is an important membrane spanning domain as well. So this one next to S4 here, this S5, this is the portion that lines the actual pore. So this is the portion that is closest. So these S5 portions here are closest to the actual pore, basically, of all of these membrane spanning domains. Okay, and that's pretty much the structure of the voltage-gated sodium channel done. All that I want to say next is that there are 9 different types known of voltage-gated sodium channels, and they all pretty much have this same, all have this same structure, where you have 4 domains of the alpha subunit, and each of these domains has these 6 membrane spanning portions. But they do have slight structural differences. They are encoded by different genes, these different voltage-gated sodium channels, but effectively they do the same thing. The only difference is that different drugs interact at them because they do have slightly different structures. You know, they have a different combination of amino acids in this polypeptide chain, so that means that, different drugs can bind to them. But they all effectively do the same thing, and they do. Another difference that they might have is, they might have different activation. They might have different threshold potentials, i.e. they might not require the cell to get as high or as low. They might require different levels of depolarization in order to open. Okay, so there are 9 different types, and they're called the voltage-gated sodium channel 1.1 to 1.9. So you have the voltage-gated sodium channel 1.1, the voltage-gated sodium channel 1.2, 1.3, 1.4, etc., all the way up to 1.9. There are 9 different sorts, and one which I specifically want to mention because it's important in it's believed to be important in inherited forms of neuropathic pain, is the voltage-gated sodium channel 1.7. So when if neuropathic pain, basically, I just want to give a little bit of an introduction about what neuropathic pain is. Neuropathic pain is when someone feels as though they are in pain. They feel pain even though there's absolutely nothing wrong with their limbs. So, nothing wrong with them, basically. So, for instance, if I had a continuous ache in my arm, even though there is absolutely nothing wrong with the arm, that's called neuropathic pain. And basically, the cause of it is that the pain neurons are firing even though there is no painful stimulus. So basically, usually what happens if someone punches you in the arm, then loads of molecules are going to be released. There's going to be a chemical storm, and all of those molecules that signal that there's been damage to that area of the arm are going to activate the pain neurons in that portion of the limb, and those will send up a painful stimulus to the brain. In neuropathic pain, what is believed to happen is that the neurons are firing, the pain neurons are firing, even without the chemical storm that tells you that, that the limb is actually damaged. And specifically, this voltage-gated sodium channel is believed to be in the dorsal root ganglia. So the cells, the cells that actually activate pain in the so let me just explain. So you have sensory neurons, pain receptors basically, that go out to your arm. So let's say let's just draw a vertebral column here. Oh dear, like this kind of thing. Okay, so there is supposed to be a vertebrae basically, and what you have is, let's say you have your arm down here, and in that arm you have neurons that are receptive to pain, okay, and those go all the way up to enter your spinal cord, and they have their cell bodies in the dorsal root ganglia, which are around here, and then they send their message on, and it synapses usually, it crosses in the midline, and then synapses, sorry, no, it doesn't, it synapses first, it synapses in, what's that lamina called? The substantia gelatinosa. It's like, it's a certain rexed lamina, I can't remember which one, but it synapses down here, and then another neuron carries it up to the brain basically. But if these sensory neurons, if these pain neurons basically, they have this voltage-gated sodium channel, this NAV 1.7, they have this certain type of voltage-gated sodium channel, and certain mutations in this channel mean that its threshold potential is greatly reduced. So usually what happens is, we know that that the neuron needs a certain amount of stimulation. You need to put in a certain amount of initial positive charge and take the electrical potential difference across the membrane to about -40. So usually, if we draw the membrane ending here, so this is the sensory ending, usually the membrane is being kept at around -65 millivolts. To make an action potential fire, what you have to do is you have to depolarize this membrane generally to -40 millivolts, and then what will happen is that the voltage-gated sodium channels, which are in this case NAV 1.7 sodium channels, those will open and allow in sodium to move into the cell, which will then trigger the upwards part of the action potential basically. Now, if certain inherited mutations in this channel structure mean that it will activate at more negative electrical potential differences, so let's say -50 millivolts maybe, and basically a chemical -50 millivolts is easier to achieve than -40 millivolts. So you need less sodium to come into the cell. You need less depolarization of this cell membrane in order to activate this voltage-gated sodium channel and send an action potential. And that's believed to play a big role in certain forms of inherited neuropathic pain, because that will mean that these neurons fire potentially unnecessarily and create painful stimuli for the brain to feel.